Thank you very much, and thank you to the scientific committee of the Congress to giving me a second opportunity to try to convince you that in rectal route we do have some choice of pharmaceutical drug dosage forms. We are not obliged to be limited to the fatty suppository. So this will be a talk, a kind of summary of two different research projects. The first one financed by Grand Challenges of Canada, and the second one by Wellcome Trust. They were both managed by World Health Organization, and all the work that I'm presenting here is pharmaceutical development, which was done at the Bordeaux University. So just a few words about the general context of this project. So according to World Health Organization, there are 16,000 children under 5 dying every day. These are the numbers of 2015. And almost the half of these deaths occurs in the first 28 days of the life. It's a neonatal period, which is the most risky. Most of these deaths occur in developing countries. If we look at the causes of this neonatal death, you can see, I'm sorry, there's a problem of spelling here. You can see that neonatal infections are important reason of these deaths. And according still to the World Health Organization, these deaths could be prevented if there were some pediatric formulations available and adapted to the local problems, meaning tropical zones, meaning easy to administer and available locally. I will talk about azithromycin and ceftriaxone that I introduced previously. So these were the two antibiotics which were chosen for their properties, antimicrobial properties, but which have quite different physicochemical properties that formulation needed to cope with. So azithromycin is a macrolide, while ceftriaxone is cephalosporin. Regarding the biopharmaceutics classification system, azithromycin is not water-soluble, but the ceftriaxone is. And concerning formulation which are available, azithromycin is available as an oral formulation in addition to injectable one, whereas ceftriaxone is available only as injectable preparation. The drug dosage form I will introduce today will include soluble suppositories, PEG suppositories. Then I will talk a bit about solid drug dosage forms, including hard shell capsules and rectodispersible tablet. And then I will end with some semi-solid or liquid formulations like self-emulsifying drug delivery systems, oily suspensions, or gels. Starting with the azithromycin PEG suppository. So this approach was only done with azithromycin because, as mentioned previously, ceftriaxone was not compatible with PEGs. An initial screening was done on different formulations of azithromycin for rectal drug delivery, and azithromycin PEG suppository appeared as the most potent drug delivery form from bioavailability in rabbits. However, this formulation was done initially as a suspension, as a solid dispersion, but where azithromycin was suspended in melted PEGs, which came along with some homogeneity issues and which we supposed had a slower drug release profile that it could have if it was dissolved. We tried to dissolve at the technical parameters used for this preparation the azithromycin in melted PEG, but it was not possible, even changing PEG chains or using a co-solvent. So this was the reason why we went on and we tried to make co-melting And the second approach was to try to find technical parameters who would allow us to prepare a solid solution. The problem of co-melting, as stated here, is that the temperature of the melting point of azithromycin was at 123 degrees, which means that we needed to work at approximately 130 degrees to make it, which represented a risk of degradation for azithromycin and PEGs. Furthermore, for this preparation, the water bath equipment could not be considered because of the temperature. And the last but not the least point was that once cooled down, the co-melted preparation, when it was melted again, there was a fine precipitate, meaning that we did not form a solution. We screened technical parameters and we found out that between 80 and 95 degrees we could form a solution, which meant that with optimal temperature at 90 degrees there was much less risk to degrade either excipients or the active principle, that usage of water bath was still possible. And when this preparation was cooled down and melted again, no precipitate was observed. So we further characterize these solid solution suppositories. X-ray diffraction here showed that here, compared to the physical mixtures, in red, which is the solid suppository, the solid suppository did not present the crystalline structure. The crystalline structure is present here in physical mixture. These peaks are due to ceftriaxone. I didn't overlay all the conditions because it would be irreadable, but these are the peaks of PEG and these are the peaks of ceftriaxone, and these peaks are missing in solid solution suppository. So we transformed the azithromycin into its amorphous state. The second point was that infrared here results showed that there was an interaction between azithromycin and PEGs, and there was a disruption of hydrogen bond between azithromycin and the water. Azithromycin in its solid state is a 2-hydrate, so there is a water included. And furthermore, differential scanning calorimetry allowed us to demonstrate that the melting point of this formulation was finally a bit lower than individual melting points, which let us think that what we formulated, in fact, was a eutectic system. We were interested in enhancement of the drug release in vitro and in vivo, and after encouraging in vitro results, we went on in vivo for in vivo bioavailability in rabbits. So here you can see azithromycin plasma profiles. And as you can see here in the table, the solid solution suppository did, in terms of absolute bioavailability compared to intravenous one. Thank you. Solid solution suppository increased remarkably the bioavailability here compared to intravenous or compared to oilless suspension, which was our rectal control condition. So much about suppositories. I'm moving on solid forms. The first form I'll talk about is a hard shell capsule. I think you all know the advantages of manufacturing hard shell capsules. I'm not going into details for that. However, when these hard shell capsules are developed for the rectal administration, there are some limits that need to be taken into account. One limit is the small quantity of the liquid available when we are administering a drug by rectal route. The second point specific to the rectal route is that the size of the capsule will be of great importance for the local tolerance. because we know that the size, the bigger the size, the more is the risk to go for a defecation reflex. Furthermore, I mean, this is even more important for small babies, which is the case here. And the third point is that at industrial scale, we would rather make sure that the flowability of the content would be acceptable. Here is a summary of our results of bioavailability in rabbits. So if I take azithromycin, which was formulated as a powder in the hard gelatin capsule, the capsule did as well as our control condition, which was the oily suspension here. In absolute bioavailability, it resulted in 13%, but did much less well than the suppository that I presented before. For the ceftriaxone, we tested granulates in the hard gelatin capsule and the powder. The powder did not flow well. This is the reason why we tested both. It could still be prepared locally in developing countries, in health facilities like hospitals, but it could not be prepared probably at industrial scale. And we can see that, at least for the powder, we approach what was our target condition, which was a fatty suppository. Another solid rectal form is a rectodispersible tablet that was studied for both APIs. Well, the rectodispersible tablets today do not exist on the market. So what we did is we tried to develop a neurodispersible-like tablet, and we tried to adapt it to the rectal route, meaning that we joined some mucoadhesive agents, and we try to work on the formulation to make sure that the disintegration would occur properly in a very small quantity of liquid. The advantage of this form, rectodispersible tablets, were regarding hard gelatin capsule, would be a decreased volume of the final form because it is compressed. In terms of formulation, we included, beside ceftriaxone or azithromycin, an absorption enhancer for ceftriaxone, a surfactant for azithromycin, and here you can see common excipients like fillers, binders, disintegrant, lubricants, which are commonly used for instance, oral drug tablet formulations. The specific point was just to find the right balance between these, for instance, between the mucoadhesive and disintegrant and the binder, to make sure that the drug dosage and the drug release will be appropriate in a small quantity of water. These are the results from rectodispersible tablets of ceftriaxone, and as you can see here from the plasma profiles, the two tablets tested here did as well as the capsule of ceftriaxone, and that the pharmacokinetic parameters were not significantly different. A rapid and significant absorption occurred in all cases. So the last part, I would like to mention oily suspensions and some gels, aqueous gels, as an example of azithromycin liquid and semi-solid forms. This is a summary also from rabbit pharmacokinetic bioavailability results. For azithromycin, we tested an oily dispersion and two different aqueous gels, which that all ended up in a comparable absolute bioavailability. So for azithromycin, the absorption was comparable regardless the medium of the preparation, oily or here aqueous. We can see, however, for HPMC gel that there is a delayed absorption here from time to reach plasma peak, which is significantly lower than the others, possibly due to its higher mucoadhesive effect. If we compare these results to the results that I presented earlier for dry formulations, azithromycin in dry formulations was as well absorbed as in liquid formulations. In terms of numbers, the numbers were even a bit higher. The standard deviation makes the difference. The difference is not significant. To end up with a small remark, I'm not going into details for this one. It was a self-emulsifying drug delivery system, which was only applied on ceftriaxone development, but would of course be of interest even more for azithromycin. So for those who are perhaps less familiar with the system, the aim was to formulate here an oily vehicle with surfactants that would, after administration, in presence of aqueous fluid, would form an emulsion. So these results are presented in a poster, so you're welcome to see me and to get some details if you're interested in. So to conclude, I hope I gave you some examples of other drug dosage forms other than a fatty suppository, which is the form that everybody thinks of when you're talking about rectal route. So in a summary, PEG suppositories for azithromycin allowed us to increase the bioavailability. Solid drug dosage forms allowed to have a significant absorption of azithromycin or ceftriaxone. And our fears at the beginning that introducing in the rectal route a large amount of the quantity of a large amount of the powder were not funded because the absorption was rapid and occurred properly. And then, according to the physicochemical properties, some other formulations such as gels, oil suspensions, or self-emulsifying drug delivery systems can be of interest. I'd like to thank people who worked on these projects. Still, Oxford University and Tide University for their help for clinically relevant choices, and World Health Organization for the managing the project. Thank you for your attention.
Yeah, thank you very much for your talk, and we have now time for a few questions. Yeah.
About this melting point of the solid dilute capsules. Melting point is a very important one in the rectal delivery system.
Yeah.
Is a melting point of this hard gelatin capsules. It should be meltable at body temperature. Can you highlight
the melting point of hard gelatin capsules?
Yeah.
They don't melt, they dissolve. Perhaps I didn't understand the question. The hard gelatin capsules would be opened in the contact with the rectal liquid, and then just release the content. As ceftriaxone, for instance, was highly hydrosoluble, the ceftriaxone dissolves in the liquid, and then in the presence of absorption enhancer, which is included in the mixture, gets absorbed.
Did you compare with these PEG suppository systems? This other one. Oil, yeah.
I'm sorry. So you would like to compare hard gelatin capsules with
this oily suppositories, PEG based.
Oily suspension?
Yeah.
With the oily suspension, the oily suspension, it depends on which active principle we are talking about. The oily suspension, here. So the oily suspension here was comparable. Sorry, it doesn't work. For azithromycin, we had comparable results for the hard gelatin capsules and the oily suspension. This is the relative bioavailability, meaning that the bioavailability of hard shell capsules was compared to oily suspension. And 107% regarding standard deviation is not statistically significant. So it's statistically comparable. It's the same. For the ceftriaxone, we had a bit higher percentages for oily solution. This is the part I didn't develop because it's on the poster. However, if I go back to capriol dispersion, this was our best condition, which was much higher than our target. So bioavailability. So the relative bioavailability, which is of 178%, was much higher than with the hard gelatin capsule, but only for this specific solvent. Well, not solvent, medium. Mm.
Okay, thank you. Is there another question? No? Then I would like to give you your second certificate.
Oh, thank you.
Oh, we need a second.